Dominance. In each community, all the species are not equally important. There are relatively only a few of these, which determine the nature of the community. These few species exert a major controlling influence on the community. Such species are known as dominants. Dominance may be due to number, biomass, or energy flow. Trophic structure, self-sufficiency. Nutritionally, each community, a group of autotrophic plants as well as heterotrophic animals, exists as a self-sufficient, perfectly balanced assemblage of organisms. Each community has its own composition, structure, and developmental history. Composition. Communities may be large or small. Larger ones extend over areas of several thousands of square kilometers, as forests. Others such as deserts, etc., are comparatively smaller with dimensions in hundreds of kilometers, and still others such as meadows, rivers, ponds, rocky plateaus, etc., occupying a more restricted area. Very small-sized communities are the groups of microorganisms in such microhabitats as leaf surface, fallen log, litter, soil, etc. The number of species and population abundance in communities vary greatly. In each community there are diverse species. All these species are not equally important, but these are only a few overtopping species which by their bulk and growth modify the habitat and control the growth of other species of the community, thus forming a sort of characteristic nucleus in the community. These species are called the dominants. Generally, in most of the communities, only a single species, due to being particularly conspicuous, is dominant, and in such case the community is called by the name of dominant species, for example, spruce forest community. In other communities, there may be more than one dominance, as in oak hickory forest community. Structure. Besides composition and dominance, the communities exhibit a structure or recognizable pattern in the spatial arrangement of their members. Thus, structurally, a community may be divided horizontally into subcommunities, which are units of homogeneous life form and ecological relation. This horizontal division constitutes the zonation in the community. Figure 1 shows zonation of different distinct vegetational types on a mountain. Latitudinal as well as altitudinal zonations of vegetation have also been shown in relation with climatic factors. In shallow ponds, zonation is very little. However, in deep ponds and lakes, there may be recognized three zones. Figure 2 is littoral zone, limnetic zone, and profundal zone. In each zone, organisms differ from each other. Another aspect of structure that is more common is stratification, which involves vertical rather than horizontal changes within the community. In each horizontal zone, there may be recognized distinct vertical stories. Sometimes the stratification is very complex, where a community possesses a number of vertical layers of species, each made up of a characteristic growth form. In grassland communities, there is a subterranean floor, containing basal portions of the vegetation such as rhizomes of grass covered by litter and debris of plants as well as animals, and herbaceous substratum consisting of upper parts of the grasses and herbs with a characteristic fauna. However, stratification in a forest community, figure 3, is most complicated, where as many as 5 vertical subdivisions may be recognized. These vertical subdivisions are I subterranean subdivision, E forest floor, E herbaceous vegetation, I peat shrubs, and peat trees. In some tropical rainforests, there may be as many as 8 vertical strata. Thus, based upon the light and relative humidity requirements, we find stratification in above ground parts. Similar stratification may also be found in underground parts, roots, rhizome, or other structures below the soil. Origin and development. A community with its particular environment constitutes an entity which has its origin and development. The details of community development will be given in the next section, ecological succession. However, for the present purpose, a brief outline of the process is being given here. In a barren area, there reach the seeds and propagules of the species. This is known as migration. The seed, as a propagules after germination, develop into the seedlings, which then develop into adults. But only a few of these survive, as by this time the new environment does not suit to all of them, and thus only a few are capable of successful growth, and this process of seedling establishment and successful growth is called esosis. As a result of migration and subsequent esosis, species colonize the new area's colonization. By this time, with the changing environment due to plants' growth, several other species of both plants and animals start colonizing the area, and sooner or later the area is colonized by a definite community. This community constitutes the biotic environment of that particular area. For quite some time, species live together in a community under a particular set of environmental conditions. But time and place affect the living organism very much, as a result of which there are chiefly two major types of relationships between the organisms of a community and their environment. These are as follows. Interrelations between organisms themselves. Such relations are chiefly in respect of food and space. These include competition. In the process of rapid colonization, individuals become aggregated at a place and develop inter as well as interspecific competition for space and food. As a result of these actions and interactions on the habitat, the pioneer species find environment. Those which are unable to adapt themselves to the changing environmental conditions disappear from the area. This changed habitat is now colonized by newer species. This community may lead to another, entirely different type of community. This orderly change, which leads to the development of vegetation, is called succession. Sooner or later this change comes to a stop, when a particular community comes to stay in both community and the habitat have reached a stabilization. This final community is called the Kumax community. Stratification. The vertical strata in a community have already been explained. For example, in forests we find several stories or layers of plant species, and in each layer plant species differ in size, form, behavior, etc. Various plants in each stratum are adapted to that particular set of local conditions, and moreover, the plants of one layer modify the environment, which is suitable for species in another layer of the community. Stratification thus is the result of interdependencies of species, as for example, lianas and epiphytes grow on other plants. Cohabitation. There are reports of a number of chemicals secreted by vegetative organs of plants of a community which modify the adaptive conditions. Similarly, chemicals secreted by fungi, bacteria, and actinomycetes in soil affect the root system as well as shoots of higher plants. Relationships between species and the environment. Organisms in the environment interact with each other and bring about several modifications. This is one of the chief reasons that a particular community changes to another type sooner or later in a particular area. Fluctuation. Once the population completes their growth, population density tends to fluctuate above and below the steady state level, carrying capacity even in population subject to various forms of feedback control. Often the fluctuation results form seasonal or annual changes in availability of resources, or they may be stochastic random. However, some population oscillates so regularly that they can be classified as cyclic. Seasonal fluctuation in population size, largely controlled by life history adaptations coupled with seasonal changes in the environment factors. E.g., at certain times of year mosquito will be abundant. Annual fluctuation may be considered as under two subheadings. I. Fluctuation controlled primarily by annual differences in extrinsic factors such as temperature and rainfall. E. Intrinsic factors. Oscillation is controlled primarily by population dynamics, biotic factors such as food or energy availability, disease, etc. Year-to-year changes in the abundance seems clearly correlated with variation in one or more major extrinsic limiting factors are called cyclic fluctuation. Characters used in community structure. It is pointed out above that each community is characterized by its species diversity, growth forms and structure, dominance, successional trend, etc. To study the details of these aspects of any community, there are taken into consideration a number of characters parameters. These are then used to express the characteristics of a community. For instance, if we say that this particular species is dominant in the community, what may be the possible characters on the basis of which such a conclusion can be drawn? These various characters used for such purposes are broadly classified into two major categories. I. Analytical characters. These are generally expressed in terms of 5-point scale, and synthetic characters are actually computed from analytical characters. Analytical characters. These are quantitative, which are expressed in quantitative terms, and qualitative, expressed only in qualitative way, although finally grouped under 5-point scale. Quantitative characters. These include such characters as frequency, density, abundance, cover and basal area, etc. Frequency. Various species of the community are recorded by different phytosociological methods, by taking any sampling unit like quadrat, transect, and point. Details of these methods will be described later. Frequency is the number of sampling units as percent in which a particular species occurs. Thus, frequency of each species is calculated as follows. Frequency percent equals no of quadrates in which species occur at x 100 total no of sampling units studied. After determining the percentage frequency of each species, various species are distributed among 1 years, 1934, 5 frequency classes depending upon their frequency values as follows. Frequency percent frequency class 0 to 20A. 21 to 40B. 41 to 60C. 61 to 80D. 81 to 100E. Density. Frequency does not give correct idea of the distribution of any species, unless it is correlated with other characters, such as density, etc. Density represents the numerical strength of a species in the community. The number of individuals of a species in any unit area is its density. Density gives an idea of degree of competition. It is calculated as follows. Density equals total no. Of individuals of a species in all the sampling units, total no. Of sampling units studied. The value thus obtained is then expressed as number of individuals per unit area. Abundance. This is the number of individuals of any species per sampling unit of occurrence. It is calculated as follows. Abundance equals total no. Of individuals of a species in all the sampling units, total no. Of sampling units studied. But abundance thus obtained in quantitative terms gives little idea of the distribution of the species. Cover and basal area, quantitative values of frequency, density, etc., do not give correct information on the distribution of a species. Since different species differ in their growth forms, such values are to be supplemented with other characters like cover and basal area. Cover, although sometimes used in general sense for the area occupied by a plant, which may be the herbage cover or the cover or basal area, it is generally used for above ground parts. Thus, cover or herbage cover signifies primarily the area of ground occupied by the above ground parts of plants, such as leaves, stems, and inflorescences viewed from above. It is a good measure of the herbage availability. It is estimated by chart quadrat, line interceptor point frame. Basal area refers to the ground actually penetrated by the stems and is readily seen when the leaves and stems are clipped to the ground surface. It is one of the chief characteristics to determine dominance. It is measured either 2.5 centimeters above ground or actually on the ground level. It is measured by calipers, line interception, or point-centered quadrat method. Since plants differ in their growth form, the relationship between herbage cover and basal area differs in different types of plants. Based on their cover values, species are grouped into 6 groups as follows. Group 1 less than 5%, 2 5 to 25, 3 25 minus 50%, 4 50 minus 75%, 5 75 minus 95%, and 6 95 minus 100%. Ron Blanket 1932 included only 5 groups. Dominance. Impact. Dominance is a synthetic character. However, Dobemeyer 1959 considered it as an analytical character and expressed its 6 types. Impact. The number of organisms sometimes may not give correct idea of the species. If we base our conclusion on number, a single or few trees in a grassland or few grasses in a forest should be of little value. But if we consider the species on the basis of area occupied or weight biomass, this situation may be different. Thus, in dominance, cover is included as important character. The single tree in grassland may occupy fairly large area and may have much mass. Qualitative characters. These include physiognomy, phenology, stratification, abundance, sociability or gregariousness, vitality and vigor, life form, growth form, etc., most of which are described and may be grouped in point scales. Physiognomy. This is the general appearance of vegetation as determined by the growth form of dominant species. Such a characteristic appearance can be expressed by single term. For example, a symbol of such a community of plants where larger trees are dominant with some shrubs would tell that it is a forest. Similarity on the basis of apoornis, it may be grassland, desert, etc. Phenology. It also includes periodicity, aspect, etc. Phenology is a scientific study of seasonal change, i.e. the periodic phenomena of organisms in relation to their climate. Different species have different periods of seed germination, vegetative growth, flowering and fruiting, leaf fall, seed and fruit dispersal, etc. Such data for individual species are recorded. A study of the date and time of these events is phenology. In other words, phenology is a calendar of events in the life history of the plant. These events are shown by phenograms. Thus, phenology of different species may differ from each other in a community. We find species with different phenology, changing the composition of community. Stratification. As pointed elsewhere, stratification is the way in which plants of different species are arranged in different vertical layers in order to make full use of the available physical and physiological requirements. Data on stratification are obtained by using a bisect. Vertical projection is constructed in stature of the species may be plotted to find out distribution pattern of stem, roots, etc. of different species. Abundance. Although related with density, abundance may not be expressed generally in quantitative terms. Organisms, particularly plants, are not found uniformly distributed in an area. They are found in smaller patches or groups, differing in number at each place. Abundance is divided in 5 arbitrary groups, depending upon the number of plants. The groups are very rare, rare, common, frequent, and very much frequent. Sociability. It denotes the proximity of plants to one another. Plants generally grow as isolated individuals, and grow better when near to each other and produce thick population. Others become weak or die in such an association. Fruit and seed dispersal, etc. and nature of reproduction are affected by the way in which they are aggregated. Species with same density may differ in sociability values. Thus, sociability expresses the degree of association between species. Some divide the sociability into as many as 10 classes, but Braun, Blanket 1932 used only 5 sociability groups as follows. S1 plants stems found quite separately from each other, thus growing singly. S2 a group of 4 to 6 plants at one place, S3 many smaller scattered groups at one place, S4 several bigger groups of many plants at one place, S5 a large group occupying larger area. Vitality. This is the capacity of normal growth and reproduction, which are important for successful survival of a species. The vitality depends upon weight of species. In plant, stem height, root length, leaf area, leaf number, number and weight of flowers, fruits, seeds, etc. determine the vitality. On this basis, plants are grouped into different vitality groups. R. Nisra and G. S. Pearl, 1954, gave 3 groups as well-developed plants completing their life cycles regularly, producing viable seeds, E. Plants reproducing vegetatively, and E. Ephemerals, short-lived. Ron Blanket, 1932, gave 4 groups. Dobemeyer, 1968, gave vitality groups as follows. B1 plants whose seedlings die. B2 seedlings grow, but unable to reproduce. B3 reproduce only vegetatively. B4 reproduce sexually, but uncommon. B5 reproduce sexually and grow regularly. Life form, growth form. In past, ecologists use the general appearance of plant communities as determined by the stature of the plant species, their spread and characters of life forms, as the basis of the study of communities. These methods are known as physiognomic methods. Of these, Ronquier's 1934 life form method has been commonly employed. A life form is the sum of the adaptation of the plant to climate. He considered the way in which different species overcome the adverse environmental conditions determines their limits of distribution. Thus, the plant's climate can be expressed by the statistical distribution of life forms in the flora of a particular region. On the basis of the position of foreign buds on plants and the degree of their protection during adverse conditions, Ronquier classified plants into 5 broad life form categories, which are shown in Figure 4. These are as follows. Phanerophytes. Their buds are naked or covered with scale and are situated high up on the plant. These life forms include trees, shrubs, and climbers generally common in tropical climates. Depending upon the size, they may be further subdivided into megaphanerophytes over 30 meters high, mesophanerophytes 8 minus 30 meters high, microphanerophytes 2 minus 8 meters high, and nanophanerophytes under 2 meters. Apophytes are either included in phanerophytes or sometimes included under a separate life form. Camophytes. Their buds are situated close to the ground surface. They are common at high altitudes. Example: Trifolium repens. Hemicryptophytes. These are mostly found in cold temperate zone. Their buds are hidden under soil surface, protected by the soil itself. Their shoot early die each year. Examples: most of the biennial and perennial herbs. Cryptophytes or geophytes. Their buds are completely hidden in the soil, as bulbs and rhizomes. Most of them are found in arid zones. Hydrophytes are the cryptophytes whose buds are found below the water surface. Theraphytes. These are seasonal plants, completing their life cycle in a single favorable season, and remain dormant throughout the rest unfavorable period of year in the form of seeds. They are common in deserts. Ron Blanket 1951, on the basis of Ronquier's life forms, divided plants into as many as 10 categories. Synthetic characters. These are determined after computing the data on the quantitative and qualitative characters of the community. For comparing the vegetation of different areas, community comparison needs the calculation of their synthetic characters. These are determined in terms of presence and constants. It expresses the extent of occurrence of the individuals of a particular species in the community. How uniformly a species occurs in a number of stands of the same type of community. The species on the basis of its percentage frequency may belong to any of the following 5 presence classes. Rare present in 1 to 20% of the sampling units, seldom present in 21 to 40% of the sampling units, often present in 41 minus 60% of the sampling units. Mostly present in 61 minus 80% of the sampling units, constantly present in 81 minus 100% of the sampling units. Fidelity. This is the degree with which a species is restricted in distribution to one kind of community. Such species are sometimes known as indicators. The species have been grouped into 5 fidelity classes as follows. Fidelity 1 plants appearing accidentally. Fidelity 2 indifferent plants may occur in any community. Fidelity 3 occur in many communities, but predominant in one, prefer ants. Fidelity 4 specially present in one community but may occasionally occur in either community as well. Selectives. Fidelity 5 occur only in one particular community and not in others. Exclusives. Dominance. It has been described under quantitative characters. Here, the dominance is expressed in synthetic form. On the basis of density, frequency and dominance covered values, there has been proposed the idea of importance value index IBI. IBI of a species in a community gives the idea of its relative importance. For IBI, values of relative density, relative frequency and relative dominance cover basis are obtained as follows. Relative frequency equals frequency of the species x 100 total frequency of all the species. Relative density equals density of the species x LO total density of all the species. Relative dominance equals dominance of the species x 100. Total dominance of all the species. Now for IBI, 3 values are added. IBI values of different species are then arranged in decreasing order. Other synthetic characters, besides the above mentioned ones, there have also been proposed some other characters. These have been quite useful in comparative studies on communities. Such characters include interspecific association and association index, index of similarity, dominance index, species diversity, and diversity index, etc. It is beyond the scope of the text of the book to go into details of such characters. F. Ecological succession, types, mechanisms, changes involved in succession, concept of climax. Ecological succession. Environment is always kept unchanging over a period of tone due to variation in climatic and physiographic factors and the activities of species of communities themselves. These influences bring about marked changes in the dominance of existing community, which in turn sooner or later replaced by another community at the same place. This process continues as successive communities develop one after another over the same area, until the final community again becomes more or less stable for a period of time. This occurrence of relatively different sequence of communities over a period of time in the same area is known as ecological succession. 1. Trends of succession. 1. Orderly continuous change in kind of plants and animals. 2. Tends to increase in diversity of species. 3. An increase in organic matter and biomass supported by the available energy flow, reverse in heterotrophic succession. 4. Decrease in net community productivity or annual yield. 2 types of succession. Autotrophic versus heterotrophic. Autotrophic succession. Succession in which most of the organic matter is fixed by autotrophs. Most living biomass is in plants. Heterotrophic degradative succession. Succession on a degradable source. Most living biomass is animal, fungal, or microbial material. Primary versus secondary. Primary succession glossary. Sequence of communities developing in a newly exposed habitat devoid of life, e.g. Succession on bare rock. Newly deposited sand. Secondary succession. Different glossary. Sequence of communities taking place on sites that have already supported life, e.g. Old field succession. Clear-cut forests, burned areas, etc. Perhaps the most useful distinction between primary and secondary succession is that the latter originates with soil. Autogenic versus allogenic, heterogenic. In autogenic succession, environment is modified by species of community itself such that conditions become unfavorable for that community and more favorable for upcoming communities. In allogenic succession, replacement of community is due to other external factors. 3 mechanisms of succession. A. Facilitation. The organisms at a given successional stage make the environment more suitable for later success stages. Examples: lichens breaking down rock into soil, nitrogen fixing plants improve fertility of soil, nurse plants. B. Tolerance. The organisms of a given successional stage have little impact on later successional stages. Example: old field succession. Possibly species of all stages get started at the same time, but are dominant at different times because of different life histories. C. Inhibition. The organisms at a given stage resist invasion by organisms of later stages. Succession proceeds when the individuals of a given stage die. Example: allelopathy. 4. Climax theories. Monoclimax theory. Climax. All vegetation within a region will converge to the same vegetation type, which is regulated by homeostasis. Polyclimax theory. Translate. There are a number of types of climax within a region, each of which reflects a different soil type, local climate, etc. Climax pattern theory. Whittaker. Succession will result in a continuum of climax types, varying along environmental gradients. Non-equilibrium hypotheses. Climax rarely, if ever, occurs. Change is continual. History and random events are important. Even if equilibrium is theoretically possible, disturbance is too frequent to allow it. Shifting mosaic steady state. Although the vast majority of sites in a landscape are changing, recovering from disturbance, the landscape may be in a steady state. This is because there is, through time, a reasonably constant portion of the landscape in each successional stage. A shifting mosaic steady state is a special case of a dynamic equilibrium. 5. General process of succession. I. Nudation. This is development of a bare area without any form of life. This area may develop due to a number of causes such as landslide, erosion, deposition, or other catastrophic agency. The cause of nudation may be A. Topographic. Due to soil erosion by gravity, water or wind, the existing community may disappear. Other causes may be sea position of sand, etc., landslide, volcanic activity, and other factors. B. Climatic. Glaciers, dry period, hails and storm, frost, fire, etc., may also destroy the community. C. Biotic. Man is most important, responsible for destruction of forests, grasslands for industry, agriculture, housing, etc. Other factors are disease epidemics due to fungi, viruses, etc., which destroy the whole population. E. Invasion. This is the successful establishment of a species in a bare area. The species actually reaches this new site from any other area. This whole process is completed in following 3 successive stages. A. Migration. Dispersal. Flesh sheets, spores, or other propagules of the species reach the bare area. This process, known as migration, is generally brought about by air, water, etc. B. Esosis. Establishment after reaching a new area. The process of successful establishment of a species, as a result of adjustment with the conditions prevailing there, is known as esosis. In plants, after migration, seeds or propagules germinate, seedlings grow, and adults start to reproduce. Only a few of them are capable of doing this under primitive harsh conditions, and thus most of them disappear. Thus, as a result of esosis, the individuals of species become established in the area circa. Aggregation. After esosis, as a result of reproduction, the individuals of the species increase in number, and they come close to each other. This process is known as aggregation. E. Competition and coaction. After aggregation of a large number of individuals of the species at a limited place, there develops competition inter as well as interspecific, mainly for space and nutrition. Individuals of the species affect each other's life in various ways, and this is called coaction. The species, if unable to compete with other species, if present, would be discarded. To withstand competition, reproductive capacity, wide ecological amplitude, etc., are of much help to the species. IV. Reaction. This is the most important stage in succession. The mechanism of the modification of the environment through the influence of living organisms on it is known as reaction. As a result of reactions, changes take place in soil, water, light conditions, temperature, etc. of the environment. Due to all these, the environment is modified, becoming unsuitable for the existing community, which sooner or later is replaced by another community, seral community. The whole sequence of communities that replaces one another in a given area is called a sera, and various communities constituting the sera. As seral communities, seral stages are developmental stages. The pioneers are likely to have low nutrient requirements, more dynamic and able to take minerals in comparatively more complex forms. They are small size and make less demand from environment. B. Stabilization climax. Finally, there occurs a stage in the process when the final terminal community becomes more or less stabilized for a longer period of time and it can maintain itself in equilibrium with the climate of the area. This final community is not replaced and is known as climax community, and the stage is climax stage. 6. Hydrosert succession in water body. 1. Phytoplankton stage, blue-green algae, diatoms, bacteria. 2. Rooted submerged stage, hydrilla, Balasnaria, Potamogeddon, Utricularia. 3. Rooted floating stage, Trappa, Nymphia, Lemna. 4. Reed swamp stage, Typha, Sagittaria, Phragmites. 5. Sedge meadow stage, Cypress, Carex, Juncus. 6. Woodland stage, Salix, Populus, Almus. 7. Forest stage, Almus, Acer, Quercus. 7. Lithosert succession on barren rock. 1. Crustus lichen stage, Rhizocarpon, Rhyndina, Lechinora. 2. Foliose lichen stage, Parmelia, Dermatocarpon. 3. Moss stage, Polytrichum, Tortula, Brynia. 4. Herb stage, Ardicida, Poa, Festula. 5. Shrub stage, Ruse, Phytocarpus. 6. Forest stage, Mesophytic trees. 8. Heterotrophic microbial, microsert succession. It begins on habitat-rich in organic matter, autogenic succession. It involves opposite progression, i.e. high energy content, more organic matter, to nil energy primary colonizer feeds on senescent tissues. Stage 1 of weak parasites, e.g. bacteria. Stage 1 primary saprophytic sugar fungi, living on sugars and carbon compounds, e.g. mucoraceous phycomycetes. Secondary colonizers feeds on dead tissues. Stage 2. Cellulose decomposers and associated secondary saprophytic sugar fungi sharing products of cellulose decomposition, e.g. ascomycetes and some mucorals. Stage 3. Lignin decomposers and associated fungi, e.g. Basidiomycetes. Climax concept in succession. The final, tenual and more or less stabilized community in succession that is able to establish some sort of equilibrium with the environmental conditions of that area was termed climax by Clemens 1916. The subject of climax has been much controversial and is reviewed from time to time by many ecologists. According to Clemens 1916, 1935, climax has the following three principal characteristics. Unity. Climax is a unit, an index of the climate area. Life or growth forms of plants indicate the climate type. Unless all the species are not taken as an organized unit, climax would not indicate the climate. Stability. The form of climax community is more or less stable with the climate. This climax community cannot be replaced through competition by any other group of species. In other words, according to Clemens, in a particular climate area there may develop climax communities only with a few characteristic dominant species. Origin and phylogenetic relations. Climax community is to be treated equivalent to an organism, like which it takes birth, grows and develops and becomes mature. In this organism concept of climax community, Clemens went to the extent of designating it a superorganism. In any climatic region, the developmental stages of the climax community have their own characteristics, which reflect the type of climate. Similar to the development of organisms with changing age, climax communities have also simultaneously undergone changes with changing climate. Thus, phylogenetic relations may be established between different climax communities of the world. There have been three popular theories about the climax concept in ecology. Monoclimax theory. As evident from the above account, F. E. Clemens emphasized the importance of only climate in the stabilization of the climax community. According to the monoclimax theory, within a given region all land surfaces eventually tend to be occupied by a single kind of community which is climax. The climax is determined by the regional climate. Given a stable climate, the climax community is stable indefinitely. However, his views were strongly criticized by many ecologists. Kells, disagreeing with the idea of stability, stated that equilibrium state is never reached and succession is in fact a variable approaching a variable rather than a constant. According to Cooper, all progressive as well as retrogressive changes in communities reflect succession. Thus he considered climax state a stage of minimum change, rather than finally changed state of succession. Climax may not be taken equivalent to an organism, from which it differs in several respects. Therefore, community may not be considered as a unit. It is common to observe that in the same climate, climax communities are different. Because, depending upon their primary stages and habitat characteristics, their successional stages are also different. Also, in same climate, the lithosert and hydrosert beginning with different pioneer communities and proceeding through different successive developmental stages might develop into similar climax communities. Similarly, it may not always be true that successions with similar pioneer communities and sterile communities would develop into similar climax communities. The most objectionable point and subject of controversy about the climax with Clemens' viewpoint has been its intimate relationship with the climate. In many conditions, in an area of uniform climate, it is common to observe different types of climax communities according to the soil, topography and other factors. Under such situations, it would not be correct to consider only the climate as determining factor in climax. For example, Hult 1885, 1887 in Finland, long before the Clemens concept of climax, described as many as 7 different habitat types under similar climatic conditions. Tansley 1935, too, disagreeing with Clemens, put forth his concept that climax communities are in fact controlled by more than one factor and not solely by climate. In this way, the climatic climax concept of Clemens became popularly known as monoclimax theory, although Clemens did not use this term. Polyclimax theory. It is indicated above how the climatic climax concept of Clemens was strongly opposed by ecologists including Tansley 1935 who believed that climax is controlled by many and not one climate factors. Accordingly, Tansley's concept became popularly known as polyclimax theory. Clemens, although agreed with the possible control of factors other than climate on climax, but he thought that these communities would sooner or later develop into climatic climax types. Consequently, with an attempt to accommodate these stages in his own hypothesis, Clemens introduced in literature a number of new terms. These are as follows. The stage in succession just preceding the climatic climax community was called a subclimax. The community, which became stabilized at any of the several stages of succession due to microclimate, or effects of factors of soil, fire, etc., was called a seroclimax. A disclimax disturbed climax was called the community which became more or less stabilized due to recurrent disturbance by man or other biotic factors, which prevent the establishment of climatic climax community in the area. At the same place, under similar climatic conditions, at smaller distances, due to differences in water and soil characters, there may develop different climax communities. Under such circumstances, Clemens 1936 coined two more terms, preclimax. It is a community with life forms lower than those in the expected climatic climax, which might develop in a habitat drier than that of the expected climatic climax, and post-climax, a strip of community with life forms higher than those in the expected climatic climax and which develops under more moist or colder habitats. As a result, followers of Clemens coined a number of new terms. For instance, coclimax, superclimax, quasiclimax, paraclimax, anticlimax, conclimax, pentaclimax, metaclimax, pseudoclimax, etc. The only way to come out of the jargons of Clemens' terminology was the polyclimax theory, Tansley, 1935, 1939. There is evidence that even under primeval conditions it was difficult to find large areas of uniform vegetation. According to this theory, the climax stage may be controlled by any factor of the environment and not only by climate. Accordingly, the climax stage is to be named, depending upon the nature of the factor in stabilization. Thus, in addition to climatic climax, controlled by climate, there may develop adaptive climax. On an underdeveloped soil, it develops due to adaptive effects. Biotic climax, develops due to biotic disturbances. Some ecologists make further categorization into anthropogenic climax due to man-grazing climax due to grazing effects and zootic climax due to animals. Topographic climax, due to differences in topographic factors of mountains, hills, mounds, etc. Fire climax, due to repeated effects of fire. According to Dobbenmeyer, 1968, if one accepts monoclimax theory, it means to admit that other factors of the environment are of secondary value. Climax pattern hypothesis. This concept about climax was put by R. H. Whitaker, 1953, who rejected the classification approach. On the basis of community gradient analysis, it was postulated that the communities developing in a particular place are in accordance with all the factors of environment. In this area, the vegetation may not be divided into smaller basal units. In fact, different climax types are orderly organized parallel to the environmental gradient according to this hypothesis. There is, in a sense, only one big community that changes according to soil, slope, and other habitat factors. Biome. As pointed out earlier in the discussion on concept of climax, in certain regions, besides the climax community, there are also associated some other communities in earlier stages of succession as well as communities of a different type, controlled by special local conditions, soil and topographic conditions. Climax communities are different for different geographical areas. In 1939, E. Clemens and B. D. Shelfer described the biome system for dealing with the geographical distribution of communities. They defined biome as biotic community of geographical extent characterized by distinctiveness in the life forms of the important climax species. The biome is not based on the species of plants and animals present, but rather it is based on the life form of the most important plants, i.e. trees, grasses, shrubs that give to the landscape its special character. The biome is viewed as a biotic community which, in this sense, means that the plants and animals are considered together as an interacting unit that gives to it a special look. The chief character of a biome is depicted by climax communities with their dominant life forms. Several communities within the region of a biome are considered part of that biome. Thus biomes, a bigger unit than community, constitute the great regions of the world distinguished on an ecological basis, such as tundra biomes, forest biomes, grasslands and deserts, etc. G1. The concept of ecosystem. 1. Preliminary definitions. In any natural system, two major components can be distinguished. A set of physical and chemical conditions, relatively homogeneous in a given geographic area, at a time t climatic conditions, for example, clearly varying according to the hours of the day and the seasons, which constitutes the biotope. A set of physical and chemical conditions, relatively homogeneous in a given geographic area, at a time t climatic conditions, for example, clearly varying according to the hours of the day and the seasons, which constitutes the biotope. A certain number of living things inhabiting this biotope: bacteria, fungi, plants, animals, which we call biocenos. For the ecologist, the basic unit of the biocenos is a population, which is defined as a set of organisms belonging to the same species. A settlement is a set of populations. There is some ambiguity between the terms settlement and biocenos. In current usage, the term biocenos is reserved for the group of organisms that habit a biotope, whereas the term settlement specifies the nature of the species considered. We therefore talk of, for example, a settlement of herbivorous mammals, Gramineae, or ichthyophagous fish. 2. The ecosystem a network of interactions. From these definitions, we could attempt, as have numerous authors since Tansley, 1935, to define ecosystem by the following equation. Ecosystem equals biotope plus biocenos. This formula now seems totally wrong, as can be seen from the following example, admittedly a caricature of a zoological park in northern Europe. For this park, a meteorological station can provide year-round data on climatic conditions such as wind speed and direction, air temperature, and rainfall. These constitute the parameters of a biotope, which we call abiotic parameters, because they are not influenced by the living things inhabiting the system. On the basis of these measurements, we observe that the temperature in winter is minus 20 degrees Celsius, that the wind blows at 100 kilometers per hour, and that precipitations occur for several months in the form of snow. Using a guide to identify fauna, we can easily identify the following species in this park: hyena, antelope, giraffe, elephant, lion, and rhinoceros. Does this mean that such animals normally live in a geographic area in which the temperature is minus 20 degrees Celsius and in which it snows in winter, since we have not observed any seasonal migration among them? Obviously not. In fact, the error introduced by the equation given above arises from the mathematical sign plus. Ever since we learned to count, we have known this sign and known that 1 plus 1 equals 2, whereas that is not the case in an ecosystem, which cannot be reduced to the simple addition of a biotope to a biocenos. In the case of the zoological park, we know very well that these animals, mostly of African origin, subsist only with human intervention. The park managers place them in heated cages during winter and feed them artificially throughout the year. As Margolef 1975 has remarked, if we were to open the cages in a zoo, the biocenos would very rapidly be simplified, being limited to one lion, obese from overeating but destined to die of hunger, and one rhinoceros, which is inedible. Left to itself, the biocenos of a zoological park is not viable, unlike a natural biocenos. The only difference between the two types of communities lies in the artificial input of matter, feed, and energy, calorific and chemical, by humans in the case of a zoo. In a natural environment, the biocenos has only solar energy and local food resources at its disposal. Because of the imposition of multiple interactions between biotope and biocenos, detailed in the rest of this work, and the network of biocenos among organisms, a functional system or ecosystem can be described as in the diagram in figure 1. 3. Concept of ecocomplexes. The idea of the functional network of interactions is in fact found in living matter at all levels of organization. A cell is not simply the addition of macromolecules. An organism is not the sum of cells or organs. A population is not just the juxtaposition of individuals that have no relation with each other or with neighboring populations. The ecosystem is not, moreover, the highest level of integration known in nature. Ecosystems are in effect largely interdependent. For example, a lake is an ecosystem of stagnant water and the slope basin that surrounds it. On the basin, two types of ecosystems can be distinguished. Terrestrial ecosystems: prairies, forests, moors, etc. One or more aquatic ecosystems of running water, one or several affluent rivers. The various ecosystems found in a territory are not entirely independent of one another. Interactions also occur between them in their physical and chemical factors. Similarly, many species are not limited to a single ecosystem and their lives may depend on different environments that are more or less far apart. In each territory, there is a set of interdependent ecosystems, exchanging matter and living organisms with one another. Such sets of interdependent ecosystems exchanging matter and living organisms between them is called an ecocomplex. Landon and Lamotte, 1988. In fact, by the pressure of movements of air and water caused by physical processes that will be described later, and because of movements that most living things are capable of migration, transport of pollen, seeds and fruits, all ecosystems are closely interrelated in the vast network called the biosphere. The biosphere can be defined as the space of the planet covering all living things and in which life is always possible. The term therefore covers, partly, the three major compartments that make up the Earth: the lithosphere, the hydrosphere, and the atmosphere. G2. Trophic levels, structure of community. Trophic levels are the feeding position in a food chain, the sequence of consumption and energy transfer through the environment. 1. Producers' first trophic level are mostly photosynthetic autotrophs, self-feeders such as plants and cyanobacteria, that extract energy from sunlight and chemical building blocks from the inorganic world, e.g., water, carbon dioxide gas, etc. 2. Consumers' secondary trophic level are organisms that consume live organic matter and transform it to ensure the constitution and functioning of their own organs. They are conventionally divided into three major categories: herbivores, carnivores, and parasites. A. Herbivores. They are primary consumers, C1, as they consume plants. They make up the second level of the food chain. Their size varies widely, from the small organisms that nibble on algae, to most insects and rodents, to giraffes and elephants. B. Carnivores. They are consumers at several degrees. Carnivores most often capture prey. By definition, they are therefore predators. They thus transform animal biomass. According to the degree of predation, we have consumers of herbivores are secondary consumers, C2, for example, a mole that eats a beetle grub, a lion that captures a gazelle, or an owl that eats a field mouse. Consumers of carnivores are tertiary consumers, C3, for example, an adder that eats a shrew, which is an insectivore. As they ingest predators, they are often called super predators. It is possible to distinguish trophic levels of the fourth, fifth, even sixth order, especially in the marine environment, where food chains are long. The same animal may belong to several levels. A buzzard is a simple C2 when it feeds on crickets, which it very commonly does in the summer. It becomes AC3 when it captures a mole or a shrew, and it is AC4 when it catches an adder, which also consumes insectivores. C. Parasites. Parasites run along the food chain. A raptor, which is AC4, may carry ticks, parasite aprids that suck its blood. These arthropods are therefore C5. But the ticks may also be victims of a superparasitism because they often harbor pathogenic bacteria or virus. That is how they transmit pyroplasmosis to their hosts. The bacteria, which are thus C6, may in turn be subject to viral parasitism by bacteriopages. These viruses are C7. 3. The detritivores and decomposers are specific consumers. It is not clear how organisms that slowly decompose cadavers and plant waste should be categorized. As they use dead organic matter to ensure their metabolism, growth, and reproduction, they behave as consumers. The result of their action is the mineralization of various elements. They thus play a significant role in the cycling of matter, which allows them to recover, in passing, the energy they need to live. This cycling is done in two stages and involves two categories of living things. Detritivores, which feed on the remains of dead organisms fallen on the soil, and decomposers, which complete the transformation begun by the detritivores in the process of mineralization that makes organic matter into mineral matter. A. Detritivores. As their name suggests, detritivores represent all individuals that feed on animal or plant detritus. Their main action, which is the first step in the transformation of dead organic matter, is the fragmentation of debris into finer elements that other transformers find in their excrement or in fecal pellets. There are several categories of detritivores according to the place or type of consumption. NECROPHAGES feed only on animal corpses. This is the case of necrophores, coleopteran insects often found on carcasses of birds, mammals, or other animals after a few days, along with other adult insects or with fly maggots. COPROPHAGES are organisms that eat excrement. The example of dung beetles, coleopteran insects, is well known. SAPROPHAGES consume decomposing plant elements. They include woodlice, terrestrial crustaceans, which find their food under rotting bark. Geophages are soil animals that play an essential role in humidification. The best known are the earthworms, which eat their way through the soil. While doing this, they digest the fragments of plant matter buried and are fallen on the soil. B. Decomposers. Once the first stage of degradation of dead organic matter is completed by detritivores, the microscopic organisms bacteria, fungi, or protozoa accomplish the second stage of this transformation. These microorganisms are responsible for actual mineralization. By aerobic processes in some cases and by anaerobic processes in others, they trigger fermentation. Fermentation breaks down the organic molecules still present in the soil in the form of glucids, protids or lipids, such as humic acids, into much smaller mineral molecules, the mineral salts. 4. Role of detritivores and decomposers in the functioning of ecosystems. By successive stages, the joint heterotrophic metabolic activity of detritivores and decomposers brings about the remineralization of organic matter that was created in the first place by the producers and that passed through all the trophic levels of the food chain. This role is important in the functioning of ecosystems since it corresponds to the completion of the cycle of matter. It again makes available to plants the nutrients they need for photosynthetic activity. In terrestrial ecosystems, the production mineralization cycle has only a few stages, since on an average 80% of the plant biomass produced is not consumed by herbivores. Such is not the case in aquatic ecosystems, where most of the plant biomass produced by unicellular algae is ingested after it is formed. In the soil, detritivores and decomposers play an often silent role in the functioning of ecosystems, that of initiating new food chains. While mineralizing organic matter, they produce their own biomass by heterotrophy. They are preyed upon by several predators that are part of the trophic network of soil. Finally, the cycling that they accomplish is essential for soil fertility. We will see in the next chapter that this mineralization may happen sooner or later and that it contributes to the proper functioning of what is called the argilohumic complex. G3. Ecological pyramids. Ecological pyramids are the graphical representation of trophic structure and function at successive trophic level of an ecosystem which may be shown in terms of their number, biomass or energy content. 1. Pyramid of numbers. To construct a pyramid of numbers, the number of individuals present in each trophic link of a given environment is calculated, in a prairie in the example described in figure. On paper, the organization looks like a triangle pointing upward. Each level is drawn as a rectangle the size of which is proportionate to the number of individuals calculated at that level. All the rectangles have the same height, only the base changes. The longer the chains, the more pointed and tall the pyramid. This imaginary representation enables us to understand some elementary principles at a glance. The number of individuals. When we go upward from the base of a trophic system to its summit, we observe that there are progressively fewer individuals in the predator chains from one link to the next. The largest rectangle corresponds to the primary producers P1. There are a large number of plants, especially in a prairie, even though there are few species. The population of herbivores C1 is much smaller, which is expressed in a smaller rectangle. That of carnivores C2 is still smaller and that of higher predators C3 is reduced to a few units. The size of individuals. Although the pyramid of numbers does not show the size of individuals, it is obvious that the reduction of numbers, as one goes up the pyramid, is compensated by an increase in size. Generally, the herbivores are larger than the grasses they consume and the carnivores are larger than their prey. Prolificity. The grasses have a Prolificity. The grasses have a tremendous power of reproduction, since each plant can produce a profusion of grains. The grasshoppers that consume them, in a very simple imaginary chain, lay only a few hundred eggs. The amphibians that eat grasshoppers in turn produce a few tadpoles. The reptiles that consume amphibians have only a few descendants. As for the black-breasted snake eagle, which eats only reptiles, it lays only one egg a year and its period of sexual immaturity is very long, 3 to 5 years. This allows us to understand why predators are the first animals that tend to disappear in cases of disequilibrium. The study of pyramids of numbers, because it does not show directly the criteria of mass and size and emphasizes only the number of individuals, must be complemented by other pyramids. Note: the pyramid of numbers for parasites is the inverse of that for predators. The number of parasites that infest a host is most of the time very high. For example, when a dog has fleas, it generally has many fleas. It is the same for lice infesting human heads. These parasites are themselves victims of hyperparasitism by acarids or viruses. Here also, the numbers of parasites are higher than those of their hosts. Thus, the inverted pyramid has narrow bottom, the primary host, and a very wide top, the viruses. 2. Pyramid of biomass. A. In terrestrial ecosystems, for this other representation of biocenosis, we draw a pyramid in which the superposed rectangles have a surface proportionate to the biomass of individuals present in each link. For grassland and forest, it is upright. For parasites, contrary to the preceding representation, the pyramid of biomass is not inverted. Even though there are many in relation to the host, the parasites have a low biomass. Thousands of fleas have a negligible weight in relation to that of the dog that carries them. B. In aquatic ecosystems, even though phytoplankton has a rate of renewal higher than that of the zooplankton that feeds In aquatic ecosystems, even though phytoplankton has a rate of renewal higher than that of the zooplankton that feeds on it, the level of primary producers base of the pyramid is narrower than that of the herbivores zooplankton. The rest of the pyramid has a usual shape fig. The phytoplankton is in fact consumed to the extent that it is produced. Its biomass therefore does not accumulate. In the sea, this is frequently the case because the zooplankton consumes all the phytoplankton produced in a day. The same phenomenon occurs in the lakes at the end of spring, where it assists in the proliferation of zooplankton. This overconsumption thus leads to a decrease in the turbidity of the water, called the clear water period. The pyramid of biomass is thus more useful than that of numbers. All the same, the pyramid does not take into account the presence of water in the tissues. For the same dry weight, plants are much more hydrated than animals. It is also necessary to calculate the biomass in dry matter and not in living matter. 3. Pyramid of energies. The pyramid of energies is the most satisfactory representation because it is the only one that really indicates the metabolic losses. According to the laws of thermodynamics, it has the same shape as the pyramids of numbers and biomass. It is upright fig. To construct this pyramid, the biomass of tissues must be converted into energy values. Generally, the unit used is the kilojoule. Sometimes, the older measure, kilocalorie, is also used, and 1 kilocalorie is equivalent to 4.18 kilojoules. 4. Decomposers and their place in the pyramids. What is lacking in the schematic representation of Elton is the place of detritivores and decomposers. It must be noted that their biomass is lower than that of primary and secondary producers, which develop living organic matter. Still, they have a very high metabolism. Figure shows that, for silver springs, the decomposers have an energy potential of 460 kilocalories, higher than that of herbivores, which have an energy potential of only 383 kilocalories. Much more than their quantitative importance is the role of decomposers in the buckling of the biomass cycle, which is the capital in the framework of the ecological pyramids. The primary producers to create their own matter must draw from the soil the mineral salts located around their roots. But these nutrients are not inexhaustible. The proof of this is the enormous quantity of fertilizers that farmers must use to improve their soils and replace the soil minerals used up by harvests. All living beings in the ecosystem, by their metabolism, eliminate a significant quantity of energy dissipated in the form of heat. When primary producers and various consumers die, their carcasses are exploited by detritivores and decomposers. Ultimately, the carcasses are mineralized in the form of mineral salts that are again absorbed by plants. This turnover is considerable if one limits it to energy references. 5. Biological magnification. It is the tendency of pollutants to become concentrated in successive trophic levels. Often, this is to the detriment of the organisms in which these materials concentrate, since the pollutants are often toxic. In order for a pollutant to biomagnify, the following conditions must be met. The pollutant must be long-lived. The pollutant must be concentrated by the producers. The pollutant must be fat-soluble. Biomagnification occurs when organisms at the bottom of the food chain concentrate the material above its concentration in the surrounding soil or water. Producers, as we saw earlier, take in inorganic nutrients from their surroundings. The problem comes up when a pollutant, such as DDT or mercury, is present in the environment. Chemically, these pollutants resemble essential inorganic nutrients and are brought into the producer's body and stored, by mistake. This is the first step in biomagnification. The pollutant is at a higher concentration inside the producer than it is in the environment. The second stage of biomagnification occurs when the producer is eaten. Not only are they taken up by the producers, but they are absorbed and stored in the bodies of the consumers. This often occurs with pollutants soluble in fats such as DDT or PCBs. These materials are digested from the producer and move into the fat of the consumer. If the consumer is caught and eaten, its fat is digested and the pollutant moves to the fat of the new consumer. In this way, the pollutant builds up in the fatty tissues of the consumers. E4. Food chain and food web. 1. Food chains. It's a sequence of organisms through which energy and nutrients move. The general sequence of food chain is from producer to primary consumer to secondary consumer to tertiary consumer to quaternary consumer to detritus feeder. Primary consumers are herbivores. All others are meat eaters, carnivores, or both eaters, omnivores. For instance, a typical food chain in a field ecosystem might be grass, grasshopper, mouse, snake, hawk. 1.1 Grazing versus detritus food chains. Grazing food chains based on producers' photosynthesis, while detritus food chains based on detritivores, breaking detritus and wastes. Grazing food chains are important in marine and aquatic ecosystems, while detritus chains play important in forest litter. 2. Food web. The real world, of course, is more complicated than a simple food chain. Food chains are not found isolated, rather they are interconnected to form food webs. The greater number of alternative channels through which energy can flow, the greater the stability of the food web in the ecosystem. The food webs you see here are grazing food chains since at their base are producers which the herbivores then graze on. While grazing food chains are important, in nature they are outnumbered by detritus-based food chains. In detritus-based food chains, decomposers are at the base of the food chain and sustain the carnivores which them. In terms of the weight or biomass of animals in many ecosystems, more of their body mass can be traced back to detritus than to living producers. 3. Energy flow across the food web. When energy passes from one level of production to the next in a food web, the transfer of energy always involves enormous losses. Moreover, a flow is defined for the energy that transits each of the elementary levels that make up a web pig. This notion of flow differs from that of energy yield seen earlier. Thus, at each link P1, C1, C2, C3, decomposers, the flow corresponds to the quantity of energy assimilated by the beings that make up that link. The flow takes into account not only the energy fixed in the living organic matter, but also the catabolic losses that enable this fixation. 3.1. Flow at the level of primary producers P1 for plants. The flow is equivalent to the real photosynthesis assimilated energy, which we have already defined as the gross production PB, and which is added to the energy stored in the existing biomass. In fact, apparent photosynthesis or net production PN does not take into account respiratory losses of plants R1. We can thus write PB equals PN plus R1. Flow equals net production of P1 plus respiratory losses. 3.2. Energy flow at the level of herbivorous consumers C1. Only one portion of the plant production is ingested by the herbivores, which we call Phi1, the part ingested by primary consumers. The rest is available to detritivores and decomposers when the plants die. As it is not used by the herbivores, we call it nu1. What is consumed Phi1 is not absorbed totally by digestion. Only a part is assimilated, or A1 flow at the level of C1. The rest is rejected in the form of excrements or secretions, which is the null 1 fraction assimilated. We must also take into account respiratory catabolic losses R2. The flow at this level is if PS1 represents the energy fix which is added to the energy already stored in the biomass of herbivores. A1 equals PS1 plus R2 flow equals energy fixed by the C1 plus respiratory losses. 3.3. Flow at the level of carnivorous consumers C2. Only part of the energy fixed by the primary consumers will be used for the functioning of carnivorous organisms or secondary consumers. This part is called EdPJ2. As for the first level of consumption, many prey die of old age and their carcasses will be put to good use by decomposers. This fraction is called BU2. Only part of the biomass consumed will be assimilated A2. All that is eliminated in the feces and various secretions corresponds to NOT2. As in the preceding level, much of the energy fixed by digestion will serve for the metabolism of carnivores and will be eliminated in the form of respiratory losses R3. If PS2 represents the energy gain that is added to the existing biomass, the flow at the level of carnivores is the following: A2 equals PS2 plus R3. Flow equals energy fixed by the C2 plus respiratory losses. The process runs in the same manner that the food chain extends to the level of supercarnivores. In view of the enormous losses that appear at each trophic level, it is clear that the food chains are always short. Detritivores and decomposers intervene also in energy flow across the ecosystem. It is these organisms that recover the energy stored in all the unused matter, nu1, Nu2, and Nu3. They draw from this energy all that is necessary for their metabolism and the increase of their biomass, and like other consumers they lose energy by respiratory catabolism or fermentation road. As was explained in the preceding paragraphs, each trophic level in the food chain is the site of significant dissipation of the energy it receives in the form of heat. This implies that the structure of biocenosis is graded. The most significant of these energy losses occurs at the level of primary production. Photosynthesis uses, by transformation of photons, only 1%, on an average, of the solar radiation for formation of plant biomass. Afterwards, during passage from trophic level N to level N+1, these losses continue to occur, although at a lesser rate. There are two reasons for this. The first reason is that all the biochemical energy contained in the biomass of the trophic level is not consumed by the higher level. The second lies in the fact that because of metabolic losses, the total gross production of a given level is not available for the higher level. Any trophic relation, like that of the consumption of prey by a predator, is of an energy type. The best yields are never 100%, since yield does not go beyond 10% on average from C1 onward. We can also say that, in thermodynamic terms, ecosystems are constantly dissipating energy. G5. Energy flow in the functioning of ecosystems. In the flow of energy and inorganic nutrients through the ecosystem, a few generalizations can be made. 1. The ultimate source of energy for most ecosystems is the sun. 2. The ultimate fate of energy in ecosystems is for it to be lost as heat. 3. Energy and nutrients are passed from organism to organism through the food chain as one organism eats another. 4. Decomposers remove the last energy from the remains of organisms. 5. Inorganic nutrients are cycled, energy is not. Many forms of energy are indispensable for the functioning of ecosystems, and it is well known that photosynthesis is not the only metabolic process sensitive enough to produce organic matter from mineral elements. However, solar radiation is undoubtedly the main source of energy flow in most ecosystems. 1. Solar energy. 1.1. Spectrum of wavelength of solar radiation. Solar radiation reaching the upper levels of the atmosphere consists of wavelengths between 10 - 4 micrometers and several kilometers. But 99% of the energy is transported by wavelengths of 0.2 to 4 micrometers, and 42% is in the visible portion of the light spectrum, 0.38 - 0.75 micrometers. 1.2. Incident energy. The quantity of incident energy, measured at the top of the atmosphere, is on an average 350 with M2, or 30 × 10⁶ kilojoules per square meter per day. There are two reasons why a significant portion of this energy does not reach the Earth's surface. The first has to do with ultraviolet UV rays, of wavelength 0.2 to 0.38 micrometers, that are practically absorbed by the ozone layer O3 located at a height of 25 kilometers. Even though these UV rays represent only 9% of the total energy, the photons they associate with have high energy and alter organic molecules. That is why, if they were to completely reach the surface of the Earth, all life would be impossible. It is likely that life first appeared in the aquatic medium because UV was absorbed while passing through several millimeters depth of water. The ozone layer, which is very thin, was formed because of oxygen produced by aquatic photosynthetic organisms of the Precambrian era, algae and some bacteria, and allowed the appearance of life in terrestrial ecosystems. The second reason is that air molecules, dust, aerosols, and water drops that constitute clouds absorb and diffuse a significant amount of that light in all directions. On an average, although the values vary considerably with the altitude, only 50% of the light energy measured at the top of the atmosphere reaches the surface of the planet. The energy received is mostly reflected by the receiving surface, this fraction constituting albedo, or absorbed by that surface. Infrared rays, of wavelength 0.75 to 4 pm, are especially absorbed by water, in which they can travel only a few centimeters. That has three consequences. A warming up of the receiving environment inducing water evaporation. Movements of air and water caused by the unequal distributions of these rises in temperature as a function of altitude and season. Emission of radiation by the surface and receiving particles. This radiation generally consists of wavelengths 10 to 15 micrometers greater than those that have been absorbed. These infrared radiations, called thermics, are rapidly absorbed by the water drops in clouds and carbon dioxide in the air. The absorption results in a warming up of the atmosphere that is known as the greenhouse effect. Average energy contribution varies from 80 to 90 with M2 at the poles to 200 to 290 W per meter 2 at the equatorial regions. 2. Biological roles of light energy. 2.1. Photosynthesis. In the biosphere, most autotrophic living things, those that can synthesize their own organic matter from mineral elements and from energy, are plants. Because of their chlorophyll, these organisms capture solar energy through a chemical reaction whose universal equation is simply CO2 plus H2O HCHO plus O2 up pointing arrow. The term HCHO represents the basal element of glucose sugars. In the chemical sense of the term, this oxido reduction requires the input of 470 kilojoules per mole 12 grams of carbon assimilated or 44 grams of carbon dioxide and 32 grams of free oxygen. Nevertheless, chlorophyll does not use all the solar energy received by the surface of the plant. Overall, radiation that can be used in photosynthesis is only 21% of the total radiation hitting the surface of the Earth. 2.1.1. Absorption spectrum and spectrum of chlorophyll action. White light, visible to our eyes and emitted by light source such as the sun or an electric lamp, is composed of a spectrum of many rays whose wavelength varies from 0.38 to 0.75 micrometers. By placing a prism in a light beam, we can break up the white light into a spectrum of rays seen in a rainbow: violet, indigo, blue, green, yellow, orange, red. All these rays do not have the same efficacy to save photosynthesis. Absorption spectrum. The absorption by chlorophyll pigments is essentially concentrated in the blue part of the spectrum, between 380 and 440 nanometers with a maximum at 430 nanometers, and in the red part, between 620 and 680 nanometers with a maximum at 630 nanometers. The wavelengths corresponding to the yellow and above all the green are practically not absorbed and cross the leaves without being stopped by the chloroplasts. That is why vegetation appears mostly green to us. In fact, although chlorophyll is the only pigment to carry out photosynthesis, other pigments like keratines, xanthines, xanthophylls also favor the absorption of light energy. These pigments play the role of reflector in transmitting a part of the energy that they themselves have captured to the chlorophyll by resonance. That energy leads to the true absorption spectrum for some plant cells, which differs from that of strong chlorophyll. Thus, the absorption spectrum in naturally occurring algae shows an increase in the use of available light in the blue area of the spectrum relative to that observed for the red part. Most of the secondary pigments capture energy associated with short wavelength light. Action spectrum. Place some plants of hydrilla under a glass funnel in a petri dish full of water. The aim is to recover and measure the quantity of oxygen free in the tube by these aquatic plants. In order to do this, illuminate the hydrilla with a beam of light and successively place in its path colored filters that correspond to rays of the white light spectrum. Then count the bubbles of oxygen released during a particular period of time with each colored filter. We logically observe that we will have a maximum efficacy of photosynthesis, measured by maximum of bubbles, with the rays that have been absorbed by the chlorophyll. This corresponds to wavelengths in the 630 nanometers for red light and in the 430 nanometers for the blue light. The green rays simply cross the chlorophyll without working. G6. Transfer of energy and matter. The energy flow caused by the impact of solar radiation in the Earth's surface is the origin of two closely complementary phenomena. It enables first of all the contact between plant cells and dissolved minerals in the oceans, lakes or saline solutions of soil. This leads logically to cellular assimilation. It then ensures an input of energy needed by chlorophyll to synthesize organic matter and thus living matter. Plants absorb only about 1.2% of the sunlight that strikes them, and about a third of that energy becomes incorporated in organic molecules that another organism might eat. These organic molecules constitute the biomass of the plant. Organisms at higher trophic levels are able to incorporate about 10% of ingested biomass as biomass of their own. Thus, a herbivore that eats 10 kilograms of plant matter may gain 1 kilogram in weight. 1. Primary production. Primary production conveys the rate per unit of time at which a given quantity of organic matter is formed from mineral matter and energy. It is expressed in mass of assimilated carbon per unit of time. Primary production is carried out by autotrophs or primary producers, which constitute the first link or trophic level of what is commonly called the food chain. The best known autotrophs are plants that synthesize their matter by photosynthesis, thus from photons. These are the phototrophs. There are also autotrophic microorganisms drawing the energy needed for the synthesis from chemical reactions. These are called chemoautotrophs or chemosynthetic organisms. 2. Productivity. Most authors define productivity incorrectly as being the quantity of biomass produced per unit of surface area during a particular unit of time, which amounts to an annual net primary production. Productivity is actually evaluated by the ratio between the production during a given time and the biomass present in the habitat. Productivity equals PP. It is expressed as a unit that is the inverse of a time t-1. For plankton, for example, in an aquatic environment where the primary production is calculated in milligrams of carbon assimilated per cubic meter of water per hour, productivity is expressed as follows: productivity equals mgfc per meter free per hour. Mgfc per meter free is inverse is the rate of renewal of biomass or turnover. Rate of renewal equals BP. In the preceding example, the rate of renewal for plankton is rate of renewal equals mgfc per meter free. Mgfc per meter free per hour. The productivity of plankton is very high. When conditions are favorable, cells can divide in 1 day. The rate of renewal is therefore 1 per day. For terrestrial plants, this turnover is much lower and varies according to the organ of the plant. It is thus 1.1 per year for leaves and 0.02 per year for wood. 3. Primary production. Only a small fraction of the sunlight striking the Earth is converted to chemical energy by primary producers. That sunlight energy that is converted to chemical energy over a given period is termed primary production. 3.1 Gross primary production. Gross primary production is all of the light energy that is converted to chemical energy by producers. 3.2 Net primary production. Net primary production is all of the light energy that is converted to chemical energy and that is subsequently stored by the primary producer, i.e., the gross primary productivity minus that employed to run the primary producer's metabolism. The ratio of net primary productivity to gross primary production gives an indication of the cost of keeping the organism going, with large ratios indicative of relatively few costs, e.g., algae, approximately 50%, and smaller ratios associated with many costs, e.g., complex plants such as trees, approximately 10%. Net primary production equals gross P metabolic losses, respiration, excretion. When the conditions allowing photosynthetic activity are combined, the gross primary production is always positive. On the other hand, net production can be either positive when the synthesis of organic molecules by photosynthesis is greater than the nocturnal consumption and various metabolic losses, respiration, excretion, or negative in the contrary case. 3.3 Order of magnitude of production. In ecological literature, there is a considerable amount of data on the primary production of various types of ecosystems. These figures have an extremely limited value because they are calculated over a few hours and extrapolated to the year. Their results thus vary considerably as a function of abiotic conditions for the same ecosystem. Abiotic conditions, in terms of solar radiation, temperature, or rainfall, have highly significant interannual fluctuations. Some authors have attempted an overall mean estimate for the major types of ecosystems on the planet. In light of the inevitably approximate nature of such estimations, the authors obtain varying values. 4. Secondary production. The term secondary production applies to all biomass produced per unit of time by organisms called consumers. Consumers only transform organic matter that is synthesized by the primary producers. The consumers are thus heterotrophic organisms. 5. Energy efficiency or outputs. Energy efficiency is defined for a trophic level as the ratio between the energy released and the energy consumed, that is, the ratio between energy fixed and energy received. 5.1. Efficiency of consumption. The consumption efficiency is the ratio between the energy integrated by consumer and energy contained in the field available to the figure. For a predator, this will be that part of energy contained in a prey which will be effectively integrated. All the net production available to the consumer is not exploited. A herbivore does not use all the net production of a plant. For example, it leaves behind roots and woody stems and prefers tender shoots. A carnivore leaves behind the bones and shell, which contain energy. The consumption efficiency varies considerably according to the ecosystems. 5.2. Assimilation efficiency. Food ingested by the consumer is subjected to mechanical and chemical digestive processes during its transit through the digestive tract. Only part of that food, the nutrients, passes through the intestinal mucus into the bloodstream. The rest is not assimilated and is eliminated with the feces. The assimilation efficiency therefore is the ratio between the energy assimilated A of nutrients and energy assimilation equals AI. It varies considerably according to the nature of the consumer organisms. In the aquatic environment, when there are massive inputs of mineral elements such as nitrogen and phosphorus, the phytoplankton grows en masse, and this growth is called the flowering of water or bloom. In such conditions, the filtering zooplankton, which is integrating by filtering the biomass present, ingests much more organic matter than it needs for its metabolism. A large part of the material ingested is liberated in the form of feces that settles in the deeper zones, where it is consumed by other organisms. A large part of this feces contains phytoplankton cells that are not destroyed by the digestive enzymes of zooplankton, and thus the growth is even stimulated by nitrogen and phosphorus liberated during the enzymatic processes. Note, all that is assimilated does not contribute to the growth of the consumer's biomass. A significant part of the energy assimilated is not fixed because it is used to meet metabolic needs of respiration, excretion, or homeothermy. Because of this, we can define another form of yield, the net production efficiency, which corresponds to the ratio of fixed energy, that is net energy N over assimilated energy A. E net production equals NA. 5.3. Ecological efficiency. Ecological efficiency is the most relevant one to our discussion because it measures the yield of a trophic relation. It is commonly called ecological yield or feed efficiency or ecological growth yield. It is the only ratio that indicates the energy losses undergone when passing from one feed level to another. It thus has a considerable advantage for the purpose of understanding the functioning of biocenosis. It is defined as the ratio between energy fixed N in the net production of a consumer and energy ingested I contained in the feed consumed. E ecological equals NI. Ecological efficiency is, for example, the ratio between the net production of a predator and the net production of the prey captured. It involves the ratio between energy values and not between weights of matter. G7. Limiting factors. Abiotic and biotic regulators determine the distribution and success of living organisms. There is range of tolerance for each of the physical and biological components of environment by organisms, and when this range is passed by any factor, it becomes limiting. 1 Liebig's law of minimum. Under steady state conditions, the essential material available in amount most closely approaching the minimum needs tends to become limiting one. It is strictly applicable under steady state conditions. There should not be any factor interaction, high concentration of same substance, or action of some other factor other than minimum one may modify rate of utilization of other. E.g., utilization of strontium in place of zinc by mollusks in shell. 2 Shelford law of tolerance. Not only too little of something be a limiting factor, but also too much is in case of factors like heat, light, etc. can be limiting. Thus, organisms have an ecological maximum and minimum range in between which represents the limit of tolerance. In aquatic organisms, oxygen is generally limiting factor. In open sea, nutrients are limiting factor. Generally, nitrates and phosphates become limiting in agricultural lands fields. Reproduction is usually a critical period when environmental factors are most likely to be limiting. 3-factor compensation. Organisms in nature adapt themselves and may even modify the physical environment so as to reduce the limiting effect of temperature, light, water, etc. Such factors compensation is effective particularly at community level but also occur within species. E.g., ecotypes, locally adapted populations, races. G8. Biogeochemical cycles in nature, chemical cycling in an ecosystem. In previous sections we have emphasized the importance of transfers of matter and energy within the ecosystem. Each living organism represents potential energy. All its organic constituents, glucids, protids, and lipids, liberate a significant amount of energy when they are catalyzed through respiratory mechanisms or fermentation, while they return to the mineral state. On the other hand, mineral salts are fixed by various autotrophic living things to elaborate new cells and keep them functioning. As for heterotrophic individuals, parasites or predators, they draw the constructive elements of their molecules from their organic environment and not from their mineral environment. Thus, matter constantly passes from a mineral state to an organic form and vice versa. The producers manufacture and store organic matter while the decomposers ensure the return to the mineral state. In this gigantic and constant cycle, each element participates in its own biogeochemical cycle. We will study those of carbon, nitrogen, phosphorus, sulfur, and oxygen, which are of greatest interest, but the cycle of other elements also deserves some attention. 1. Carbon cycle. The most important of the chemical elements characterizing the living world is carbon. In the mineral form, it is present on the globe in carbon dioxide, hydrogen carbonates, and carbonates. In the organic form, it constitutes glucids, protids, and lipids. 1.1. The 3 natural reservoirs of carbon. On the global scale, there are 3 reservoirs of carbon: the atmosphere, the oceans, and the continental biosphere. Only the atmosphere contains purely mineral carbon. In the other 2 reservoirs, the 2 forms of carbon are associated: the organic form and the biomass of living things in the mineral form, and hydrogen carbonates or carbonate sediments. A. Carbon dioxide stored in the atmosphere. Stored carbon dioxide occupies around 0.03% of the volume of the atmosphere. It is estimated at 700 gigatons, 1 gt equals 1090. Its equilibrium is regulated by constant exchanges between the 3 reservoirs. B. Carbon stored in the oceans. Living things in the seas and oceans represent only a small part of the carbon that is in the waters. Most of the carbon is present in the water in the form of soluble elements. HCO3 are monohydrogen carbonates, CO2 and carbonate ions CO3 2. The carbon stored in the surface. Water is estimated at 600 gt and the water in deep seas is estimated at 39,000 gt. Over geological time, sedimentation and compaction led to the formation of carbonate rocks such as chalk or dolomites, which represent a formidable reserve of 30 million gt. C. Carbon stored in the continental biosphere. Carbon in the continental biosphere is estimated at 700 gt for living things and 1100 gt for the soil, totaling 1800 gt. In addition, there is the carbon contained in fossil fuels, such as petroleum, coal, and peat, of around 6,000 to 10,000 gt, not counting bituminous schist, which may constitute sizable reserves. 1.2. Biochemical and physiochemical exchanges between the 3 reservoirs. A. Between the atmosphere and the continental biosphere, the exchanges between these 2 sources are essentially linked to the activities of living things. The primary producers, autotrophs by photosynthesis or chemosynthesis, fix CO2 to ensure the synthesis of their organic constituents. The consumers then transfer these constituents into various food webs. Each year, by means of these biochemical exchanges, autotrophs keep up a gigantic flow of carbon of the order of 100 gt, which is extracted from the atmosphere pig. On the other hand, other biochemical exchanges balance these extractions by emitting into the atmosphere an identical flow of 100 gt of carbon, figure 89. These releases of mineral carbon result from catabolism of all living things. To carry on their various functions, these beings consume energy, which is provided by cellular respiration for animals as well as for plants, or by fermentation. In the soil, the most common mineralizers are the lactic, alcoholic, and butyric fermenting agents. These decomposers enrich the atmosphere in CO2. B. Between the atmosphere and the oceans, the flow of carbon between the air and the oceans is equally balanced and of the same order. Physiochemical exchanges at the interfaces cause a release of 100 gt of C02 into the atmosphere and an identical absorption of 100 gt of this gas by the water. The quantity of carbon dioxide dissolved in the oceans is around 50 times as much as that present in the atmosphere. Also, the entire atmospheric CO2 is renewed every 8 years by exchanges that conform to the following equilibriums between these 2 reservoirs. The first equilibrium, with reversible reactions, is established between dissolved carbon dioxide and hydrogen carbonate ions, HCO3, which may represent 90% of carbon in the oceans. CO2 plus 2H2O left right arrow H3O plus HCO3. The second is established between hydrogen carbonates and carbonate ions, CO3 to 2. HCO3 plus H2O left right arrow H3O plus CO3 to 2 calcium carbonates and magnesium carbonates, which are poorly soluble, precipitate and accumulate in the sediments. Over geological periods of time, they thus store formidable reserves of mineral carbon in the carbonate rocks. C. Human activities and carbon emissions. For our industrial, domestic, and agricultural activities, we constantly burn increasing amounts of fossil fuels, petroleum, coal, in boilers and motors. We exploit chalk, incinerating it to fabricate cement or lime. Certain crops such as rice harbor symbiotic group bacteria that are responsible for a significant emission of methane. Moreover, because of the digestive activity of our herbivorous livestock, the quantity of methane released into the air has quadrupled in the past few decades. Ultimately, human activities are responsible each year for the emission of 5 gt of CO2. Some 2 gt should be added to that for the consequences of deforestation in the tropical forests, which limits their photosynthetic role. 2/3 of this additional 7 gt of human origin are observed by the ocean and the soil. Assessment of this cycle thus makes it clear that there is an annual increase of 2 to 3 gt in atmospheric carbon. 12.1.3 Fossilization of organic carbon. In deep bogs, the activity of mineralizing microorganisms is interrupted by cold, humidity, and the acidity of the medium. The organic residues, still rich in energy, accumulate in the form of peat. In the course of geological time, an analogous mechanism of carbonification has led to formation of coal from buried, partly decomposed plants. Petroleum is similarly formed by suspension of the mineralization of marine organisms accumulated in reducing abysses. To the nitrogen cycle. Nitrogen is the element best known to farmers, who intervene actively in the nitrogen cycle by adding nitrogen fertilizer or by certain cultural practices. Mineral nitrogen is abundant on our planet, since its gaseous form, N2, atmospheric nitrogen, represents most of the air we breathe. On the other hand, nitrate ions. Note 3, the only form of mineral nitrogen that can be assimilated by plants, and ammonium ions are highly unstable and easily leached. Organic nitrogen constitutes all animal and plant cells. It also plays an important role as a metabolite, since it is found in amino acids, the basic links of all proteins. It is also involved in the construction of nucleic acids in the form of puric and pyramidal bases. When living things die, the agents of putrefaction, mostly aerobic bacteria, mineralize the organic nitrogen according to the process of nitrification, which happens in several stages big. By the mediation of symbiotic bacteria, certain plants, especially the leguminosae, can fix atmospheric nitrogen, which biochemical reactions reduce to ammoniacal nitrogen and then to proteins. 2.1 Fixation and transformation of atmospheric nitrogen. A. Fixation by symbiotic bacteria and cyanobacteria. Gaseous molecular nitrogen can be reduced to ammonia by microorganisms. These are sometimes located in the rhizosphere of roots, as with free fixers such as azotobacter or aerobic bacteria or Clostridium anaerobic bacteria. They are also found on nodules located on the roots or stems of certain floristic groups, as with symbiotic fixers such as rhizobium on leguminosae or Frankia, symbiotic bacteria of Casuarina and alder. Microorganisms can also live in the aquatic environment, such as cyanobacteria, also called cyanophyceae. Some of these organisms float by means of specialized cells, the heterocysts, and thus fix nitrogen in the air to balance their NIP ratio in an optimal way. The reduction of atmospheric nitrogen leads to a synthesis of ammonia molecules that immediately participate in the elaboration of amino acids and then proteins. N2,2-HH equals NH2HH2 and NH2,2H2 Dimide hydrazine ammonia. This reduction is catalyzed by a bacterial enzyme, nitrogenase, which is inactivated by oxygen. Also, the nodules, large consumers of oxygen, must be apportioned by the transporters, the leghemoglobins, which facilitate the diffusion of O2 under very low pressure without inactivating the nitrogenase. B. Transformation of N2 by meteorological phenomena. At high altitudes, multiple rays transform atmospheric nitrogen into NH3+ and nitrogen oxides. This also happens during storms because the colossal amount of energy liberated by lightning acts in the same way. Added to these photochemical and electric effects is the influence of certain precipitations. European farmers say that snow in February is as good as fertilizer because the slowly failing flakes capture nitrogen, which is then incorporated in the soil. 2.2 Mineralization of nitrogen. A. Nitrification. When living things die, microorganisms that are autotrophic with respect to nitrogen progressively degrade the proteins into ammoniacal mineral nitrogen, nitrous nitrogen, and finally nitric nitrogen. These three stages set off the oxidation of these various substrates by aerobic bacteria, which draw from it the energy required for chemosynthesis. The result of these redox pairs is similar to that which results from photosynthesis, that is, C3 molecules that are taken up again by various metabolisms. Here it is called chemosynthesis because the energy comes from chemical reactions and not from photons. CO2 plus 2H plus energy C3 molecules C6 molecules. Ammonification action of ammonifying bacteria. If the soil is sufficiently aerated, bacterial enzymes catalyze the oxidation of amino acids contained in the decomposing proteins which releases ammonia or NH4 plus ions. RCHNH2COOH plus O2 RH plus 2CO2 plus NH3 plus ammonia RCHNH2COOH plus O2 plus H plus RH plus 2CO2 plus NH4 plus ammonium ion. Nitrosation action of nitrous bacteria. Other aerobic autotrophic bacteria, such as Nitrosomonas europaea, use ammonia or ammonium in a redox pairing. Transform them into nitric acid or into NO2 ions. According to the following reactions. 2NH3 plus 3O2 plus 2HNO2 plus H2O plus energy. Nitration action of nitric bacteria, highly unstable nitrous acid. Or NO2 ions from nitrosation are recovered by bacteria of the nitrobacter type, which oxidize them into nitric acid or into NO3 ions. These combine quickly with soil cations to give nitrates, which the plants can then absorb. 2HNO2 plus O2 2HNO3 plus energy. 2NO2 plus O2 2NO3 plus energy. B. Denitrification. When soils are not aerated, the nitrates are lost to the plants because anaerobic bacteria metabolize them into nitrites, nitrogen oxide, or free nitrogen N2. The bacteria Pseudomonas transforms nitrates into ammonia. Nitrococcus denitrificans transforms them into nitrogen oxide. Theobacillus denitrificans, which is autotrophic with respect to sulfur and nitrogen, degrades sulfur and nitrates respectively into SO4 2 and N2. 2.3 Synthesis of fertilizers in the nitrogen cycle. Industry, by making numerous synthetic fertilizers available to farmers, intervenes to some extent in the nitrogen cycle. Many nitrogen fertilizers are in fact synthesized by reduction of the nitrogen in the air, since they must release either number 3 or NH4 plus ions. Their production triggers chemical reactions using the sulfuric acid or nitric acid. For example, H2SO4 plus 2 NH3 plus NH4, 2SO4 ammonium sulfate, HNO3 plus NH3 plus NH4 number 3 ammonium nitrate. These two fertilizers, with calcium nitrate, C8 number 3, to obtain chemically, are the most commonly used. Those that directly input nitrates act very quickly but are a significant source of pollution because they are highly soluble. Basic slow-acting fertilizers are also used, such as urea, Colorado NH2, 2 calcic cyanamide con 2. Ammonitrates simultaneously input fast-acting nitric nitrogen and ammoniacal nitrogen, which acts more slowly. 3. The phosphorus cycle. On the scale of the biosphere, the phosphorus cycle is incomplete or open. Like the other elements of biogeochemical cycles, phosphorus exists in the mineral and organic form. The stored mineral phosphorus available to biocenosis is entirely contained in the rocks. It is found in the lithosphere and apatites or fossilized sediments such as phosphorites. This relatively rare element in the biosphere has a cycle that has two phases, one that acts in terrestrial ecosystems, the other in aquatic ecosystems. Big. 3.1. Phosphorus in terrestrial ecosystems. The dissolution of phosphate rocks makes mineral carbon available to plant roots, which they can absorb and incorporate into biomass. Phosphorus is a major element in the metabolism of living things. It is especially involved in the constitution of essential molecules involved in the major functions of organisms. It is involved in the elaboration of tissues, nervous system, skeleton, and controls heredity and energy transport. Above all, phosphorus is found in the nucleic acids, DNA and the RNAs, in the phospholipids of the membranes in the brain, in the energy-rich molecules ATP, ADP, and AMP, and associated with sugars, glucose 6-phosphate, ribulose diphosphate, and other molecules. Phosphorus circulates in biocenosis through the food webs. It is first absorbed by plants and then incorporated in herbivores, carnivores, parasites, predators, and finally decomposers. When living things die, microorganisms mineralize the organic phosphorus molecules into phosphates. Some of these may be newly absorbed by plants and keep up a more or less complete cycle on land. But most phosphate ions are leached and run off into the aquatic ecosystems. 3.2 Phosphorus in aquatic ecosystems. Because of runoff, there are dissolved phosphates PO43 in fresh water and in the oceans. Phytoplankton captures and uses them in their development. In normal conditions, the phosphorus is insufficient to ensure optimal photosynthesis of algae and their development is rather limited. Sometimes, on the other hand, because of human activities, there is excessive phosphorus in the water. The disturbance of the NP ratio leads to an abnormal proliferation of certain algae or bacteria and eutrophication of waters. As in the continental biosphere, mineral phosphorus captured by the algae is incorporated in the organic form in aquatic food webs. It passes from phytoplankton to zooplankton, then concentrates in the mollusks, fish, mammals, and aquatic birds. A small part of the phosphorus in oceans returns to the land because of fishing or through the excrement of aquatic birds. Fish are one source of high amounts of phosphorus. As for bird excrement, it is collected from rock cliffs on which the birds build their nests, especially in Chile, and used as guano, a phosphate fertilizer. Another part, mineralized in the form of sediments from cadavers of aquatic creatures and accumulated in shallow water, rises by the phenomenon of upwelling ascending currents. It can also be incorporated into food webs by phytoplankton and thus goes through a closed cycle. But the greatest quantity of this mineralized phosphorus is lost in very deep sediments. It thus escapes cycling and represents the open part of the phosphorus cycle. It ultimately fossilizes in the form of phosphate rocks within the oceanic plates. 4. The sulfur cycle. The biogeochemical cycle of sulfur is best known by the phenomenon of acid rain, in which it combines with the action of nitrous oxides. Like other elements, sulfur is present in two forms, mineral and organic. The organic form appears only in the constitution of proteins that contain sulfurate amino acids, cysteine, cysteine, and methionine. In the molecules, it is characterized most often in disulfide bridges and thiol groups. The mineral form, on the other hand, is quite well represented in the atmosphere and terrestrial and aquatic ecosystems. In the air, sulfur is mostly present in the form of sulfurose or sulfur dioxide, sulfate sulfurate hydrogen H2S or sulfates SO4-2. In the water and sediments, mostly sulfides S2 are seen. In the soil it is found in minerals such as iron sulfur, pyrite, Fes, or calcium sulfate or gypsum CaSO4 from which plaster is made. The fossil fuels, coal and petroleum, also contain sulfur Pig. 4.1. Sulfur in the atmosphere. Sulfur in the air comes from various sources. The four major sources are the following. Gases emitted by volcanoes. The sulfiters release a hot water vapor that is highly charged with SO2 and above all H2S. Around the mouth of the volcano, the sulfurous gases are deposited regularly at the vent openings in the form of magnificent yellow flakes of sulfur. Combustion of hydrocarbons in coal in factories, heating of domestic stoves, and motor vehicle emissions release fumes rich in SO2. Erosion and dissolution of soils by rainwater also enriches the atmosphere in sulfurous elements. Finally, sulfur bacteria, which are autotrophic with respect to sulfur and profuse in marshes and reducing mediums that are normally hostile to life, metabolize sulfur and draw energy from it to reduce CO2 into organic molecules. They also release sulfurose anhydride or sulfurate hydrogen into the atmosphere. 4.2. Sulfur in the sediments. The largest reserve of sulfur is found in sediments. It accumulates here when conditions are reducing, combining with iron, phosphorus, calcium, magnesium, and other elements. This sedimentation is constantly being produced in marshes, but mostly in the sea bottoms, in eustenic abysses. These sediments, when compacted, constitute rock minerals such as gypsum and pyrite. In the same way, when petroleum or coal is formed over geological periods of time, sulfurate sediments are added to organic matter, which is fossilized. This explains why the combustion, if these fuels is so polluting, is partly responsible for acid rain. 5. The oxygen cycle. 5.1. Production of molecular oxygen. The oxygen cycle is closely linked to those of carbon and water since the essential source of free molecular oxygen is photosynthesis. It is because of green plants that the air we breathe contains 21% of O2 in volume. This reserve is maintained by the forest, which each year releases about 15 t of oxygen per hectare, and also by the ocean's pig. In the aquatic environment, phytoplankton plays the role of trees. It maintains an equilibrium that compensates the consumption of dissolved oxygen by biocenosis. 12.5.2. Degradation of molecular oxygen. Oxygen is consumed by respiration and fermentation, which characterize the catabolism of living things, and also by various combustions linked to human activity. The Krebs cycle of cellular oxidation is dependent on energy provided to cells in the form of ATP. Since it is a powerful oxidant, oxygen nevertheless remains toxic to organisms. Also, its obligatory cellular utilization necessitates the intervention of appropriate molecular transporters such as hemoglobin and blood or lemon globins in plants. Part of the oxygen in air is lost in the oxidation of minerals, as for iron. 4PO plus O2 2K2O3 molecular oxygen is also involved in the formation of ozone by reaction with UV rays. O2 plus O3 a small part is finally associated with carbon monoxide from volcanoes or the combustion of hydrocarbons to produce CO2. 2CO plus O2 2CO2. Note, if all the fossilized organic molecules forming over geological time were burnt, there would no longer be oxygen on Earth. 6. Hydrologic cycle or water cycle. Salt water evaporates from sun's energy producing fresh water in clouds, leaves salts in ocean. Water vapor cools and condenses to precipitation over oceans and land. Runoff forms freshwater lakes, streams, ponds, groundwater, and is held in plants and transpired. Gravity returns water to oceans, but some move slowly through water table and porous aquifers between rock layers. Although the water cycle shows water to be a renewable resource, the 3% of water that is fresh may be polluted. H. Biogeography. Major terrestrial biomes. Theory of island biogeography. Biogeographical zones of India. Biogeography is a science that documents and attempts to explain the patterns of distribution of populations, species, and ecological communities across Earth. Earth's biogeographic regions. Explaining species distributions might seem to be a simple matter, because the question of why a species is or is not found in a certain location has only a few possible answers. If a species occupies a particular area, either it evolved there, or it evolved elsewhere and dispersed to the area. If a species is not found in a particular area, either it evolved elsewhere and never dispersed to the area, or it was once present in an area but no longer lives there. Determining which of these possible answers is correct requires information about the evolutionary histories of species, which comes from fossils and from knowledge of their phylogenetic relationships. It also requires information about changes in Earth's geography, such as continental drift, glacial advances and retreats, sea level changes, and mountain building that occurred as the organisms were evolving. Such geological information can tell us whether organisms evolved where they are currently found or dispersed and colonized new areas from a distant area of origin. The biomas of the continents differ enough to allow the division of Earth into several major biogeographic regions. Biogeographic regions are based on the taxonomic similarities of the organisms living in them. The boundaries of biogeographic regions are set where species compositions change dramatically over short distances. Figure 1. The biomas of the biogeographic regions differ because oceans, mountains, deserts, and other barriers restrict the dispersal of organisms. Although there has been dispersal of organisms between adjacent biogeographic regions, such interchanges have not been frequent enough to eliminate the striking differences that have resulted from speciation and extinction within each region. A species found only within a certain region is said to be endemic to that region. Remote islands typically have distinctive endemic biomas because water barriers greatly restrict immigration. If the islands are large enough or form part of an archipelago, allopatric speciation often produces unique species and communities. For example, nearly all the trachyphytes and vertebrates of Madagascar, a large island off the eastern coast of Africa, are endemic to that island. Madagascar by itself could be called a biogeographic region, but because dozens of islands would qualify as biogeographic regions on the basis of the distinctness of their biomas, islands are treated differently from continents. Most species are confined to a single biogeographic region. The most widespread species today is probably Homo sapiens, but a few other species, for example, the great egret, the osprey, the peregrine falcon, and the barn owl, are found on all continents except Antarctica. Next, we will discuss the influence of speciation, extinction, and other historical processes on biogeographic patterns, and then consider the influence of processes operating today. History and biogeography. Before 1850, most people, including biogeographers, believed in a relatively unchanging Earth that was too young for long-term processes to account for the diversity and distribution of life. Linnaeus 1758, for example, believed that all organisms had been created in one place, which he called paradise, from which they later dispersed. Indeed, because most people believed that the continents were fixed in their positions, the only way to account for the current distributions of organisms was to invoke massive dispersal. The notion that the continents might have moved was not seriously considered until 1912, when Alfred Wegener, a German meteorologist, argued that the continents had drifted over time. Wegener based his theory on several observations. The shapes of continents, the outlines of Africa and South America seem to fit together like pieces of a puzzle. The alignment of mountain chains, rock strata, coal beds, and glacial deposits on different continents, the distributions of closely related species that were shared between Africa and South America, which were difficult to explain if the continents had always been where they are now. When Wegener proposed that the continents had moved, few scientists took him seriously. There were no known mechanisms to move continents, and no convincing geological evidence of such movements existed. Geological evidence and plausible mechanisms were eventually discovered. The broad pattern of continental movement, which continues today, is now clear. About 280 million years ago, the continents were united to form a single landmass, called Pangaea. The continents then began to separate from one another, but when the continents were still very close to one another, about 245 Maya, many groups of terrestrial and freshwater organisms, such as insects, freshwater fishes, frogs, and trachyphytes, had already evolved. The ancestors of some organisms that live on widely separated continents today were probably present on those land masses when they were part of Pangaea. By 100 Maya, continental drift had separated Pangaea into northern, Laurasia, and southern Gondwana land masses, and the southern continents were moving away from each other. Over time, India separated from Africa and slammed into southern Asia. Australia moved closer to Southeast Asia, and South America, which had drifted as an island for 60 million years, came into contact with North America. Throughout the history of life, continental drift has both separated and combined biotas, thus greatly influencing the distribution and evolution of species. Biogeographers convert phylogenies to area phylogenies. As the age of Earth, the geological processes that shaped it, and the mechanics of evolution became better understood, biogeographers were able to ask questions such as, where and when did evolutionary lineages originate? How did they spread? What do the present-day distributions of organisms tell us about their past histories? A technique that was developed to help answer these questions was the creation of area phylogenies. To generate an area phylogeny, biogeographers alter taxonomic phylogeny by replacing the names of the taxa with the names of the places where those taxa live or lived. For example, an area phylogeny suggests that horses speciated as they moved from Asia to Africa, whereas the speciation of zebras took place entirely in Africa. Figure 2. Biogeographers use several approaches to infer the approximate times of separation of taxa within a lineage. First, if a molecular clock has been ticking at a relatively constant rate, the amount of difference in the molecules of species should be strongly correlated with the length of time their lineages have been evolving independently. Second, fossils can help to show how long a taxon has been present in an area and whether its members formerly lived in areas where they are no longer found. A third valuable source of information is the distribution of living species. Much more information can be gathered on current distributions than will ever be available from fossils. Similarities in the distributions of many lineages of organisms provide clues about past events that affected them. Bickering events and dispersal both influence distributions. The appearance of a barrier that splits the range of a species is called a Bickering event. A Bickering event divides the population of a species even though no individuals have dispersed to new areas. If, however, members of a species cross an already existing barrier and establish a new population, the species' disjunct range is a result of dispersal. By studying a single evolutionary lineage, Bickering events and dispersal both influence distributions. The appearance of a barrier that splits the range of a species is called a Bickering event. A Bickering event divides the population of a species even though no individuals have dispersed to new areas. If, however, members of a species cross an already existing barrier and establish a new population, the species' disjunct range is a result of dispersal. By studying a single evolutionary lineage, a biogeographer may discover evidence suggesting that the distribution of an ancestral species was influenced by Bickering events such as a change in sea level or mountain building. See figure 3. If that inference is correct, then species in other lineages are likely to have been influenced by the same event. That is, a number of lineages may have similar distribution patterns. Differences in distribution patterns among lineages may indicate that the lineages responded differently to the same Bickering events, that the lineages separated at different times, or that the lineages have had very different dispersal histories. By analyzing such similarities and differences, biogeographers can discover the relative roles of Bickering events and dispersal in determining today's distribution patterns. The longer an area has been isolated from other areas by a Bickering event, such as continental drift, the more endemic taxa it is likely to have, because there has been more time for evolutionary divergence to take place. Australia, which has been separated the longest from the other continents, about 65 million years, has the most distinctive biota of any continent. South America has the next most distinctive biota, having been isolated from other continents for nearly 60 million years. North America and Eurasia, which were joined together for much of Earth's history, have very similar biotas. That is why the early European travelers felt more at home in North America than in Australia. When several hypotheses can explain a pattern, scientists typically prefer the most parsimonious one, the one that requires the smallest number of unobserved events to account for it. We saw how the parsimony principle is used in the reconstruction of phylogenies. To see how it is applied to biogeography, consider the distribution of the New Zealand flightless weevil Lycrobius hadoni, a species that is found in the mountains of South Island and on sea cliffs at the extreme southwestern corner of North Island. Figure 3. If you knew only its current distribution and the current positions of the two islands, you might surmise that, even though this weevil cannot fly, it has somehow managed to cross Cook Strait, the 25-kilometer body of water that separates the two islands. However, more than 60 other animal and plant species, including other species of flightless insects, live on both sides of Cook Strait. Although organisms do cross marine and terrestrial barriers, it is unlikely that all of these species made the same ocean crossing. In fact, we do not need to make that assumption. Geological evidence indicates that the present-day southwestern tip of North Island was formerly united with South Island. Therefore, none of the 60 species need to have made a water crossing. A single Bickering event, the separation of the northern tip of South Island from the remainder of the island by the newly formed Cook Strait, could have split all of the distributions. As we have just seen, the distributions of species today have been determined, in part, by history. However, because all organisms must be able to survive in today's environmental conditions, Earth's climates also exert a powerful influence on the distributions, abundances, and evolution of species. Ecology and biogeography. The climate of a region is the average of the atmospheric conditions, temperature, precipitation, and wind velocity found over time. Climates vary greatly from place to place on Earth, primarily because different places receive different amounts of solar energy. We will first examine how these differences in solar energy input determine atmospheric and oceanic circulation. Then we will show how climates influence the geographic distributions of organisms. Solar energy inputs drive global climates. Every place on Earth receives the same total number of hours of sunlight each year, an average of 12 hours per day, but not the same amount of energy. The rate at which solar energy arrives per unit of Earth's surface depends primarily on the angle of sunlight. If the sun is low in the sky, a given amount of solar energy is spread over a larger area and is thus less intense than if the sun is directly overhead. In addition, when the sun is low in the sky, sunlight must pass through more of Earth's atmosphere, so more of its energy is absorbed and reflected before it reaches the ground. Thus, at higher latitudes closer to the poles, there is greater variation in both day length and the angle of arriving solar energy over the course of a year than at latitudes closer to the equator. On average, mean annual air temperature decreases about 0.4 degrees Celsius for every degree of latitude, about 110 kilometers at sea level. Air temperature also decreases with elevation. As a parcel of air rises, it expands, its molecules move farther apart, its pressure and temperature drop, and it releases moisture. When a parcel of air descends, it is compressed, its pressure rises, its temperature increases, and it takes up moisture. Earth's climates are strongly influenced by global air circulation patterns, which result from the global variation in solar energy input that we have just described and from the spinning of Earth on its axis. Air rises when heated by the sun. Warm air rises in the tropics, which receive the greatest solar energy input. This rising air is replaced by air that flows toward the equator from the north and south. The coming together of these air masses produces the intertropical convergence zone. Typically, heavy rains fall in a region when it is close to the intertropical convergence zone as the rising air releases its moisture. The intertropical convergence zone shifts latitudinally with the seasons, following the shift in the zone of greatest solar energy input. This shift results in a characteristic latitudinal pattern of distribution of rainy and dry seasons in tropical and subtropical regions. Figure 4. The air that moves into the intertropical convergence zone to replace the rising air is replaced, in turn, by air from aloft that descends at roughly 30 degrees north and south latitudes after having traveled away from the equator at great heights. This air, which cooled and lost its moisture while rising at the equator, now descends, warms, and takes up rather than releasing moisture. Many of Earth's deserts, such as the Sahara and the Australian deserts, are located at these latitudes where dry air descends. At about 60 degrees north and south latitudes, air rises again and moves eastward or away from the equator. At the poles, where there is little input of solar energy, air descends. The black arrows around the edge of figure 5 show these vertical air circulation patterns. These movements of air masses are responsible, in part, for global wind patterns. The spinning of Earth on its axis also influences surface winds because Earth's velocity is rapid at the equator, where its diameter is greatest, but relatively slow close to the poles. An air mass at a particular latitude has the same velocity as Earth has at that latitude. As an air mass moves toward the equator, it confronts an increasingly faster spin, and its rotational movement is slower than that of Earth beneath it. Conversely, as an air mass moves forward, it confronts an increasingly slower spin, and it speeds up relative to Earth beneath it. Therefore, air masses moving latitudinally are deflected to the right in the northern hemisphere and to the left in the southern hemisphere. Winds blowing toward the equator from the north and south veer to become the northeast and southeast trade winds, respectively. Winds blowing away from the equator also veer and become the westerly winds that prevail at mid-latitudes. The broad arrows in figure 5 show the average directions of these surface winds. When wind patterns bring air into contact with a mountain range, the air rises to pass over the mountains, cooling as it does so. Because cool air cannot hold as much moisture as warm air, clouds frequently form and release moisture as rain or snow. On the leeward side of the mountain range, the now dry air descends, warms, and once again picks up moisture. This pattern often results in a dry area called a rain shadow on the leeward sides of mountain ranges. Figure 6. Global oceanic circulation is driven by wind patterns. The global pattern of wind circulation drives the circulation of ocean water. Ocean water generally moves in the direction of the prevailing winds. Figure 7. Winds blowing toward the equator from the northeast and southeast cause water to converge to the equator and move westward until it encounters a continental landmass. At that point, the water splits, some of it moving north and some of it moving south along continental shores. This polar movement of ocean water that has been warmed in the tropics is a major mechanism of heat transfer to high latitudes. As it moves toward the poles, the water veers right in the northern hemisphere and left in the southern hemisphere. Thus, water flowing toward the poles turns eastward until it encounters another continent and is deflected laterally along its shores. In both hemispheres, water flows toward the equator along the west sides of continents, continuing to veer right or left until it meets at the equator and flows westward again. The climates created by these atmospheric and oceanic circulation patterns play key roles in determining what kinds of organisms can live in a given region, as we'll see in the next section. Terrestrial biomes. In addition to recognizing biogeographic regions, ecologists also classify communities of organisms into biomes, ecosystem types that are based on the structure of their dominant vegetation. The vegetation of a biome has a similar appearance wherever on Earth that biome is found, but the plant species in these communities, despite their physical similarities, may not be evolutionarily closely related. Biomes are named for and identified by their characteristic vegetation, sometimes supplemented by a description of their location or climate, but each biome contains many species and all other taxonomic groups. The distribution of biomes on Earth is strongly influenced by annual patterns of temperature and rainfall. Figure 8. In some biomes, such as temperate deciduous forest, precipitation is relatively constant throughout the year.